I'll get started. I would actually be introducing Coralie Williams, but she couldn't be here today, so I am going to be her mouthpiece and present her talk today. We are going to be talking about meta-analysis, and these are some sort of practical things if you are doing a meta-analysis, things to think about and to help you out as you go through the process of a meta-analysis. So first of all, we have seen a huge increase in the number of meta-analyses published in scientific literature in the last 15 years or so. So you can see, like, even in 2010, it was relatively low, and it's just shot up considerably in the last 15 years. So it's probably good as researchers, if we're going to engage in this type of research, to be really meticulous about the choices that we make, so that we come up with a final product which is really good and really high quality. So first, we really should sort of put some context around what meta-analysis is. It is a method to synthesize results across multiple primary research studies to answer a specific research question. So you're using stuff that's already been published to create a new set of information about a particular research question. It's part of a systematic review. Well, it's only one part. It tends to be like the statistical part of a systematic review. And it provides a quantitative summary of the available evidence that's been published. And it can answer broad questions that individual studies tend not to be able to answer due to resource limitations and things. So for example, what is the effect of exercise on depression across a bunch of different populations and settings? Usually studies like that are focused on particular populations or in particular locations, or maybe the interventions are limited. Or something like, what is the effect of warming temperatures on crop yields across a bunch of regions and crops? Something like this might focus in particular regions or on just a few different types of crops. But with a meta-analysis, we can combine lots of different studies to get a better estimate overall. So this talk is going to sort of has three parts. We'll talk a little bit about systematic reviews and meta-analysis, some practical aspects like software choice, some things you should be thinking about when you're actually doing and engaging in the meta-analysis statistical part of it. And then going into some advanced topics. Hopefully I'll get there. I tend to talk a lot, so I don't know if we'll actually be able to do all of that, but that's my intent anyway. Okay, so we should provide a little bit of information about what the systematic review process is and how meta-analysis fits into that. A systematic review, as opposed to sort of a narrative review, it follows a rigorous, predefined protocol to minimize bias and maximize transparency. And there are a number of steps that you should follow as you're going through this process. Going from defining your research question, which is always something you need to do when you're doing a study. There's no difference here. All the way down to reporting results. And in meta-analysis, we're really focusing in this seminar, it's really focusing on 5 through 8. So starting with the statistical aspects where we're pooling the effect sizes, exploring heterogeneity and bias, and then reporting the results. But none of that can happen, and none of that can happen at a really high quality level if the steps before them aren't high quality as well. So you really have to pay attention and do a really nice job when you're selecting your research question, finding your studies, extracting your data, in order to get a nice, really good product at the end. One of the best tools out there to help you do that is the PRISMA guidelines. They provide a checklist, this is only part of it, that you should go through as you are doing your meta-analysis to make sure that you've done every single thing on that checklist. It's very handy and very helpful, and it keeps you on track. There are a couple of other tools as well that will help us. The Cochrane Handbook, you can read it for free online. You can also buy the book if you want, and there are also PDFs that you can download. And pre-registration of your meta-analysis in the Prospero database is a really good idea as well. So you can go check that out if you are interested in learning more. I'm going to see if I need to, yes, I need to start the air conditioning again. There we go, okay. Now, software for meta-analysis, there's lots of different things that you can choose to help you with your analysis. We have some proprietary software packages, like you can, Stata is a really great package for meta-analysis, but you have to have a Stata license. SPSS, you may have access to it through UNSW. It's only recently included modules for meta-analysis, and I don't know that it's particularly comprehensive, but it does do some meta-analyses, so it might be adequate for your purposes. You'd have to check that out. And then there are some purpose-built proprietary software, so RefMan and comprehensive meta-analysis. And there are also a number of open source tools for you as well. I haven't used most of these, but we're going to be focusing on R because it's open source, it's free, and there are lots of different packages available to do meta-analyses. And one of the most popular is the Metaphor package, and that's what we're going to be using. So now we're going to start talking about a little bit about the actual process of the meta-analysis. And you may be familiar with meta-analyses because you often see the forest plots, and that's sort of what you look at when you're looking at the results of a meta-analysis. A nice figure is always really easy to interpret, gives you a lot of information. But there's a lot of work that goes into it before you can even make that plot. Okay, so once again, our systematic review process, we were starting at number 5. So we're going to talk about some pooling effect size estimates first. Okay, so effect sizes. I think that every talk we've had in this room has talked about effect sizes, and I'm going to do it again. In the context of a meta-analysis, it's a standardized measure of the magnitude of the effect of interest in an observed study. And so common examples of these things, if you've got a continuous outcome that you're interested in summarizing across, you might be looking at means and standard deviations. And often you might be trying to estimate a group difference effect, or like, treatment versus control effect. So you'd be meta-analyzing the standardized mean difference. There are other effect sizes for continuous outcomes as well. But the most common, particularly when you're talking about a comparison, is the standardized mean difference. Lots of meta-analyses look at binary outcomes. So you've got total sample size and then the number of people or observations that experience a particular event in a group or two groups. And we can use an odds ratio or a risk ratio or even a risk difference as the effect size to summarize in a meta-analysis. Correlations, we can simply use a correlation. That's sort of a standardized measure in the first place, so that's really nice. We can do meta-analyses for time to event studies or survival analyses using the hazard ratio. And we can even do meta-analyses for count rates outcomes, so person time outcomes, by using incident rate ratios. These are just the most common ones. If you have enough information, you could do a meta-analysis on any type of a statistic, really. Now, sampling variances, this is a really important thing to think about, or a concept to understand when you are doing your meta-analysis. These describe the uncertainty around each study's effect sizes. Almost every talk that we've had talked about how you get an estimate and then there's some variability around that estimate or in our sample, and that's what we're talking about here. Statistics is really about looking at how variability is in our sample and sort of quantifying that uncertainty. So when we're doing a meta-analysis, these sampling variances are calculated from the summary statistics that are reported in the primary studies. And we need information like sample size, standard deviations, event counts if you've got the binary data. And also we can use confidence intervals in order to estimate sampling variances. You'll find if you are doing meta-analyses, not everybody is, as Lewis showed, not everybody is really good when they publish their articles and giving us the statistics that we want. You don't always get your mean and standard deviation. Sometimes you get a median and interquartile range, or you get proportions when you really should be looking at odds ratios to report a difference between two groups. So published studies are going to be sometimes very good and a lot of times not very good at giving you the information that you want. But as part of the data extraction process, you can sometimes calculate the values that you need or do conversions to get to the values that you want. And the goal in a meta-analysis is to pool those effect sizes to estimate an average effect and quantify the uncertainty amongst all of the studies that you have. And when we do that, in meta-analysis, we generally have two different ways, statistical models that we use to do this. And the first one is called the fixed effect meta-analysis. And the major assumption here is that there's one effect size really across all studies. And any variability that you see across the studies and the estimate of that effect size is sampling error. It just happened that the sample was a little bit different. So the assumption is there's one effect and it's only sampling error for the meta-analytical model. The other model is called a random effects meta-analysis. And this one says, no, effects actually do vary around a mean, and it's not just sampling error. There's some other stuff that goes around in it too. So we should actually partition the variability into that sampling error and then some other between study variance as well. So the differences amongst the study means or study effects that you have, the variability is due to sampling variability and other factors. And we separate the estimation into those two parts. So when we are pooling our estimates, those two assumptions lead to some different ways that we treat our data in the aggregation process. Now, one of the things that we're interested in and concerned about when we are aggregating across a bunch of different studies is like how much weight do we put on a particular study? And that variability estimate, or the assumptions between the fixed effect and the random effects models has consequences to how we are weighting each study in our final aggregated model. So for the fixed effect meta-analysis, our weights are only dependent on that sampling variability variance. But for the random effects, it is based on both both parts of that variability, the sampling variability and the between study variance as well. So when if you were doing a meta-analysis and you ran a fixed effect meta-analysis and a random effects meta-analysis on the same data, you are going to see that the weights of the studies in those two different analyses are quite different. And that is because of the different assumptions on where the variability amongst your effect sizes is coming from. You will also see that not only do you have slightly different values for each of the effect sizes from your studies, you can have quite a big difference in the different effect sizes in your data set. We can quantify and examine that heterogeneity. And these things can be driven by not just the sampling variability, but differences amongst the studies themselves. So they're done on different populations, they've used different study designs, and maybe even the quality of the research that itself contributes to the variability amongst the effect sizes. So we can quantify heterogeneity with a few different statistics. The most common ones are Cochrane's Q, and it measures how different each study's effect size is from the overall pooled effect size. And then we can use that information to calculate what's called I-squared, which is a value that ranges from 0 to 100%. 100% meaning that's a heck of a lot of heterogeneity, 0 meaning everything looks the same. There are some general guidelines on what's high heterogeneity and what's low heterogeneity. Now, one thing to remember when you look at your heterogeneity statistic is that just because it's high does not mean you've got a bad study meta-analysis going on. So do not feel badly if you still, you run you go through the Prospero or the checklist, and you've done a really good job of getting your data set together, and you run your analysis and you've got 90% heterogeneity. It doesn't mean that you're doing a bad meta-analysis. It just means that you've got variability amongst the effect sizes. And actually, it's a sort of a nice way to motivate a deeper exploration of what's going on in your data. So visually, you can also see on your forest plot the heterogeneity when you see that the confidence intervals, and I'm going to show you the forest plot in just a moment. The confidence intervals, if they're not overlapping. If there's not a lot of overlap, it goes along with a high heterogeneity statistic. There are a couple other ones that you can do as well. So here we're going to go through an example. These are results from 9 students on the length of studies, length of hospital stay for stroke patients, and they were either getting some sort of specialized care or conventional care, non-specialized care in the hospital. And these are the data, and we can see that we have ends for each of the 2 groups, means for the 2 groups, and standard deviations for the length of stay that is reported in each of these studies. Now, I said we were going to be using R and metaphor, so let me just point out a couple of things here for you. This command scalc is effect size calculation. It's best for you to provide the raw data that you have extracted to R, and R can do some calculations for you, so that you don't have to do all of this by hand or yourself. And that's what this scalc command does. It takes the information you've given it for your particular effect size, and ours is going to be a standardized mean difference. So we need our means for our 2 groups, our sample sizes for the 2 groups, and the standard deviations for the 2 groups. And what comes out are 2 extra things in our data set. We have this yi value and the vi value. The yi is our effect size. That's our standardized mean difference. And the vi is the variability around that estimate for each of the studies. And those are the 2 things that we're actually going to pass to metaphor for the actual meta-analysis itself. So we can see here, we're using RMA, that's a random effects meta-analysis, and we're saying, hey, take our outcome variable, which is y sub I, that's our standardized mean difference, and the variability estimate, and run a random effects meta-analysis, and then show me the results. So these are the results that we get out of it. So let me just point out a few things that are relevant and of interest to us. So here we have our tau squared. This is our between study variability, and that a random effects meta-analysis estimates. We have our I squared, which is our one of our statistics telling us about the amount of heterogeneity amongst our studies, and it's pretty high, 95.5. So we have lots of evidence of lots of heterogeneity. There's H2, another one of the heterogeneity statistics. And then we have our Q statistic down here, which also tells us that, yeah, we've got some lots of heterogeneity. And then we actually have the results down at the bottom. So our estimate of the standardized mean difference is negative 0.54 with confidence limits negative 1.14 to 0.07 and a p-value of 0.08. So after pooling results from all studies, there is no evidence of a difference in mean length of stay for the specialized care group versus the routine care group with those results right there. That's our aggregated estimate says. We do have high heterogeneity, so there may be some other things that we can do to explore why there's some such differences amongst the groups. So if we had study level information about this, then we could sort of go on and explore that as well. And this is usually what we think of when we're talking about meta-analysis. We want to see what this is, because pictures are very easy to digest and get a really nice, quick idea of what is going on. So first of all, you can see that there is quite a bit of non-overlap of our confidence intervals for each of the studies. We do have an estimate and the confidence interval of the aggregated effect on the bottom, and you can see that upper limit is above that null value of 0. But that, yeah, and so we have each of the estimates as well. So what if I squared is high? So it's often recommended to do a random effects meta-analysis if you've done a fixed effects one and you have high heterogeneity. The thing is, though, it doesn't fix anything. You still have high heterogeneity. You just also have partitioned your variability into a couple different types. But like I said, it's not a failure of your research project to have high heterogeneity. There can be very many reasons why high heterogeneity exists, and you can take a look and think about your data set in particular and what makes sense with your research question to explore what might be happening. So it could be due to differences in populations. Maybe there are, those studies are done in quite different regions, and so their populations are quite different. And so maybe if you explore something like that, you might find that does account for some of that high heterogeneity. The thing is that any pooled effect that you might get may not actually represent any of the studies in particular at all. It is mathematically and statistically a pooled effect, but it may not actually correspond to any particular study that you have in your data set. In which case, generalization can be difficult, and it is it's kind of standard to try to figure out maybe subgroup analyses or moderator or meta-regression analyses that might help explain some of that, that observed heterogeneity. Any questions before I talk a little bit about publication bias? Yes?
In the forest plots, the two studies that seem to be different than the rest, is that severity of stroke?
That is an excellent question. I don't know.
It seems like it is, that it's a high
Oh,
severity
yep,
stroke.
yep, very well could be. And that could be driving the heterogeneity statistic. Yeah, yeah, very well could be. Might be hard to really test that very much because there are only two studies, but yeah, it very well could be that.
It'd be hard to add a moderator.
Yeah, yeah. Okay, so publication bias. This is something that you should always be concerned about when you are doing meta-analysis. Because published studies tend to, everybody gets really excited about significant results. Evidence of difference or evidence of a relationship, let's publish that. They are less excited to publish null results or nothing, no evidence of anything happening. And when you combine that with the fact that small studies, so studies with small n's, are only likely to be published if there is a significant effect. Small studies with large effects are going to be the ones that are going to be published. So this is sort of, when those two things combine, when you have a small study, it tends to have a really large effect. And when you don't have lots of studies that have small effects and are not significant, it can sort of combine to get an aggregated effect that is larger than what is actually happening. So it is very important to check for possible publication bias in meta-analysis. Now, these things I'm going to be talking about do not prove that publication bias exists in your data set. It can only suggest that it is a possible reason or possibly present in your data set. Now, we do this in a number of ways, and if you are doing a meta-analysis, you should not rely on one. You should be doing multiples of these to get sort of lots of different definitions and ways to look at it. And the first one, the most common one, is a funnel plot, and it is a plot of your effect sizes versus your standard errors. And what you want to see is dots that are sort of spread evenly on either side of your effect line. So you want symmetry, and if you see asymmetry, then you may have a publication bias problem. There's your trim and fill, where you impute some hypothetical studies that may be actually missing from your data set and estimate an adjusted effect size. If it changes a lot, then you might have some problems with publication bias. Another is Egger's test when this is simply a regression of your effect sizes on your standard errors. And if there is evidence of a relationship there, then perhaps you have some publication bias. The P-curve and selection models are a bit more technical and involved, but those are good tools as well. So I'm going to go through publication bias, and then I think I might not have time for the rest of the stuff. Okay, so this is what a funnel plot looks like, and we can see that we've got this white triangle, and then we have dots everywhere. What we're really concerned about are these studies that are really far away from our effect line and have large standard errors. So those are the smaller studies. And that is a study that has a small sample size and a big effect. If you have too many of those, then and then not anything on the other side as well, so or more toward the sort of estimate line, you may have evidence of publication bias, such that smaller studies with large effect sizes are more present than small studies with smaller effect sizes. So that triangle represents the 95% confidence intervals around the overall effect size estimate. And if you're concerned that not too many of those dots are actually inside the triangle, it's okay. That happens when you have high heterogeneity, which we know we had. Okay. So for this, we do see that the high heterogeneity. We also can see that the study with the large effect has this large effect size also has a large should be large standard error. But as far as seeing any definite evidence of asymmetry, it's kind of unclear, and that may be because we only have 9 studies, and it's hard to tell a pattern with only 9 studies. So we should also pay attention to some other tests of asymmetry or tests for publication bias. We can do a correlation between the weights, effect sizes, and sampling variances, and we can see here that we get a Kendall's tau of -0.22, and that has a large p-value, so this is not suggesting that there's really any publication bias, and there's not a lot of evidence here. And Egger's test, where we do a regression of the effect sizes on the standard error. And again, it's not suggesting that this is something that's happening in these data sets. Okay, what time is it? I do have a little bit more time, so maybe I'll try to do this one. Okay. So sometimes when you are extracting your data, you find that lots of studies will have relevant effect sizes, multiples. They report multiple effect sizes that you're interested in. So you're getting more than one effect size per study. But one of the assumptions of the fixed effects and random effects models is that the effect sizes are independent. So how do you deal with the fact that you've got these effect sizes that are coming from the same study that are obviously going to be correlated with one another? So this is the situation that you have. So if we're talking about cluster, if you consider study number 1, that first study could give you 3 different effect sizes that are relevant to your research question. And that study number 2 is giving you 2. And then this last one only has 1, but depending on how many studies you have, you could have many more individual effect sizes than you do studies, because of the fact that each study might give you more than one effect. But you can still do meta-analysis. You can do what's called a multi-level meta-analysis. If any of you have done any multi-level models or mixed models, this is the same idea. What we need to do is allow heterogeneity at more than one level. So not just between studies, like we were doing before, but also within a study. These multi-level meta-analysis models allow us to do that. So these models account for the non-independence among clustered effect sizes, and allows the heterogeneity to be modeled at multiple levels, so within and between studies. And this leads to better estimates of standard errors when there's clustering, because if you do not take account of the clustering, your inference is going to be wrong because the standard error estimates are wrong. So like I said, you may have heard these talked about before: multi-level random effects models, mixed effects models, hierarchical models. So here's a quick example. These are results from 17 studies on the association between recidivism and mental health in juveniles. The outcomes are in terms of standardized mean differences, where positive values indicate higher prevalence of recidivism amongst those individuals with a mental health disorder. And you can see in the data set that we have 6 different effect sizes from that first study. Now, each of these effect sizes are relevant to a particular behavior. So they're all estimates of delinquent behavior, but different aspects of the delinquent behavior that they were studying. And you can see that study number 2 has 3 different estimates. So not surprisingly, we can use R to do this, and it's going to look really similar to what we did before. But we're using this rma.mv, that's multi-level command. And I want to point out to you this part of the code right there. This is saying, hey, we have multiple effect sizes, which was called ESID in our data set, within study. So what it's saying is that yeah, we have clustered data, and we're defining the clustering by the study ID. And in our output, looks a little bit different, and in particular, we have variance components. So rather than just having one variance estimate, we actually have 2. We have one at the study level and one at the effect size level within study. We still have our heterogeneity statistics and our results down at the bottom. So we have an estimate of 0.43 with these confidence limits and a p-value. So we do have evidence of a positive association between mental health disorders and recidivism amongst our juveniles. We do have evidence of heterogeneity and the 2 separate sort of estimates, the 2 types of heterogeneity. These are the estimates of the heterogeneity at each level. Now, you might be concerned about whether that is too complicated a model for your research question. Do you really need those terms? And you can test whether including those 2 separate types of variance estimates is a good idea by comparing that full model with one where there's sort of the study level a variance is set to 0, and seeing if there is a difference in model fit there. So we do that by including this little bit right here, where we're saying, hey, that first variance estimate, assume it's 0, and then run the model again, and then we can test the model fit between the one with both of our variance estimates. And we have a likelihood ratio test with a p-value that's very small, so we have some evidence that it's a good idea to estimate that value. And then you can do the same thing for the second estimate right there. And again, we have evidence that this is a good idea to estimate them separately. So we do need that a more complicated, less parsimonious model due to the clustering within study. There is a lot more that I could go into, but I talk a lot, and it is probably best for me to stop now and answer any questions. Yes?
Have you experimented with any other forest plot software? I find that one is great until you want to modify it even the tiniest amount.
Oh, it's you can end up with code that is this long for a simple forest plot. I've used Stata, and it can be slightly easier, but not much. I suppose there might be some like graphics programs that you could do forest plots in, and they probably look better. I mean, those are very simple, very straightforward, but you can probably make some really nice ones. I've also seen people use ggplot, but I don't know that that's any easier, because you really have to know ggplot code in order to get the diamonds and all of the stuff that you want to do out of it. So I don't know that there's I haven't found anything that's like markedly easier.
I got a question.
Okay.
Could you go back to
Well, yeah, I agree. I think that what this is mostly telling us is that there's so
Because
much
I just wonder,
variability.
as a funnel plot to this, sometimes they're telling us not anything about asymmetry or publication bias, but they're telling us we fit the wrong column.
Yeah, you definitely could interpret it that way here, because that, I mean, the biggest thing in this particular funnel plot is the heterogeneity, I think.
There's only
Yep.
really one dot that's in the drum.
Yeah, yep, yep. Yeah, I think you're probably right, there's probably a better way to analyze these data. Okay, we've got one other talk. I want to encourage people to stick around for the public lecture at 4 o'clock with Merlin Crossley. Should be very interesting. But we've got about 5 minutes, and then we'll have a talk on data visualization, I think. I just realized I did that talk without the microphone.